We'd like to get started, if we could. As is our custom, I'm going to just say a few general comments, and then I'll introduce Jerry Santacora to introduce our speaker today. But a couple of announcements. First, the upcoming Grand Rounds presentations. Next week, our Grand Round speaker is Nolan Williams from Stanford, who's not talking about whether or not naltrexone blocks the antidepressant activity of ketamine. But he will be talking about the interface of psychiatry, cardiology, neurology, and radiology, probably talking about some of his deep brain stimulation and other neurostimulation research. The following week, February 1st, Dean Alpern will be here talking about the state of the medical school and the state of the university. And then on the 8th of February, B.J. Casey from the Department of Psychology here at Yale will be talking about the emergence of the adult brain and her work on brain development, reward, cognition, circuitry in childhood and adolescence. Couple of things I want to draw people's attention to. One is that we sent around an email, forwarded an email from the provost, which is sent around to all the departments to remind people how important it is to both register your financial interests and conflicts of interest with the Conflict of Interest Committee, and to disclose all of those conflicts of interest in all of your publications. That's not news. No one should be surprised by that, but I think it's a useful reminder. The second thing is just to let people know that Bob Sherwin, who's was the founding leader of the Yale Center for Clinical Investigation, our CTSA, is retiring from that position, and they are looking for candidates who might be interested in co-leading the CTSA with Brian Smith. And so if you're interested in taking that on, you should let him know. Let's see, what else? I think that's about it. Other than to celebrate the fact that we have concluded another season of psychiatry residency interviews. We've interviewed how many people? 156. 156 of the most impressive young people that you could ever meet. And if you ever have any doubts about the vitality of the field or have concerns about being hopeful for the future of psychiatry, I can tell you that these were extraordinary people, inspiring young people, and I want to commend and particularly thank our residency directors and residency team for fielding this group of interviewees, and to thank all of you who were a part of the enormous job of interviewing these people for all the hard work. I hope you and
especially the residents who met them the night before and talked with them at lunch.
I will repeat that, especially the residents who met with them the night before and actually told them the truth about what it's like to be a resident at Yale. So at this point, I want to introduce Jerry Santacora to introduce our speaker today, but I just want to welcome Holly. I've known Holly, or I probably have known you since you were a resident, since you worked with my brother, Andrew, at Duke, before going to Columbia, then back to Duke, and now at NIMH. We've followed your career with remarkable. It's been a remarkable career filled with accomplishment, and I'll let Jerry tell them all about it.
I'll actually try to keep this relatively brief. I've actually also known Holly, I think, since your days at Duke. I don't even know, went down there for a 2-week rotation with you and Andy. So it's been 25 years ago or so. But probably didn't want me to say that. I'm sorry about that. But no, it really is my pleasure to introduce Dr. Lisanby, who has really been a champion and a leader in the development and implementation of neuromodulation technologies in the treatment of neuropsychiatric disorders. Holly's career has really spanned the breadth of doing preclinical non-human primate research up to being involved in large phase 3 trials. It actually started at Duke as an undergraduate and then stayed for your MD and residency, moved to Columbia for a fellowship, and then stayed and really developed the neuromodulation program at Columbia, then moved back to Duke, taking the position as chair of the Department of Psychiatry in 2010. And again, developing a neuromodulation program, or expanding what was a very successful ECT program into a broader neuromodulation program at Duke. And then in 2015, moving over to assume the position as the director of the Translational Research Division at the National Institute of Mental Health, which is a very important position for Holly, but also for all of you. So you should be aware of that. But then not only running that division, but Holly has also went on to really have the intramural research program in non-invasive neuromodulation, starting that unit and experimental therapeutics and pathophysiology branch. So very full docket of work for Holly. So with that, I'd like to just introduce her. She really is pushing the envelope on how we're delivering neurostimulation and neuromodulation to try to get the biggest benefit we can from these techniques with the minimal invasive methods. All right, Holly, thank you.
Thank you. Thank you, Jeffrey. Thank you. Well, thank you for the kind invitation. It's really an honor to be here at such a fantastic department that has made so many important contributions scientifically over the years and continues to do so, and of course top-rate clinical services, especially in ECT and other areas, and such an excellent training program. So it's really awesome to be here. Thank you for this opportunity. If there are any neurologists in the audience who think I'm going to be talking about how to treat epilepsy based on my talk, I'm sorry to disappoint you, but I hope you'll stay anyway. I'm going to be talking about using seizures to treat psychiatric disorders, and in particular, new advances in improving the precision in the way we deliver seizure therapy, especially sculpting the spatial distribution of the electric field in the brain and its temporal components in terms of how we deliver it. As you heard in the introduction, I also have extramural roles in terms of grant mechanisms, and not just with NIMH, but also part of the NIH Brain Initiative. And in the question and answer session or during the lunch, I'd be happy to answer questions about that. But my talk this time is going to be, during the grand rounds, is going to be about the history, present, and future of precision seizure therapy in psychiatry. These are my disclosures. I work for the federal government. And I'm part of the government that's not shut down, so I'm really happy to be here today. So why am I talking about seizure therapy? Suicide rates are increasing. This was the disturbing report from the Center for Disease Control that in both women and men over the 15-year span shown here, in 1999 in the green bars and 2014 in the red bars, suicide rates went up. This is despite our best efforts at high-quality clinical care and research to improve treatment for conditions that lead to suicide, mainly mental health conditions, severe mental illness, depression especially. Fortunately, we have very effective and rapidly acting treatments for severe depression, such as electroconvulsive therapy, or ECT, and I know that you all have a very robust and active program here, and also have been one of the leaders in studying the neurobiological actions of ECT in animal models. ECT, among our FDA-approved antidepressant treatments, is the most potent today with an effect size of 0.8. And if you compare the response rate, this is the response rates from the STAR-D trial in the, I guess it's sort of a teal color here. So after the first antidepressant medication, patients with depression, about half of them will respond. When they fail to respond to that first medication, they are switched to a second, and only less than a third will respond to that, and then they're switched again, and so on. And you can see that for treatment-resistant depression, which is down here, there's diminishing returns from medication switches. However, ECT has a very much higher, maybe 4 times higher response rate. That response rate is achieved quickly. These are data from the Prolonging Remission Depressed Elders study, which is an NIMH-sponsored study that we published. The green line here shows you the drop in the depression scores after individual ECT treatments. So this is the first treatment, a significant drop in depression score. So that's a very rapid onset of action. In our study of over 220 patients who were elderly with depression referred for ECT, we had a remission rate of 83% among completers. Fast acting, this bar graph here, the orange bars are the cumulative histogram showing you the percentage of patients achieving response. After 2 weeks, you see about half have achieved that, and after 3 weeks, close to 3/4 have achieved that. So it's rapidly acting. We know with severe depression and acutely suicidal patients, the speed of action may be the difference between life and death. And indeed, we see that ECT rapidly resolves suicidal ideation. This histogram shows the suicide item on the Hamilton Depression Rating Scale before ECT in the blue, and you see a significant percentage of patients endorsing active suicidal thoughts. After the course of ECT in red, you see that histogram shifted to the left, with the vast majority, almost all patients, having a complete resolution of their suicidal ideation. It also has an exceptionally broad spectrum of action. There's evidence of efficacy in a long list of disorders, not just depression, notably schizophrenia, the self-injurious repetitive behaviors in autism, among others, treatment-resistant status epilepticus, Parkinson's disease. So it's really broad in its action, which makes it of great interest to understand mechanisms that could lead to insights to develop novel, more targeted therapies for a broad range of psychiatric and neurological conditions. One of the neurobiological clues, and in fact this department yielded some of the first data on this topic, is that ECT is a potent inducer of neuroplasticity. In fact, a broad range of neuroplastic actions have now been reported with ECT in terms of inducing and altering a transcription of a number of genes. And then the molecules induced by that gene induction, a long list of those, and then a long series of neuroplastic effects. And I'll show you some of these, specifically cellular proliferation and mossy fiber sprouting. We conducted an NIMH-sponsored study in non-human primates to understand the neuroplastic actions of ECT, and also an experimental form of treatment, which I'll get to later in the talk, which is magnetic seizure therapy. And these are just our methods. It was a pathological study. Following acute and high doses of ECT, we found increases in mossy fiber sprouting, which is the growth of aberrant synaptic connections in the dentate gyrus, shown here, these little black patchy areas. And indeed, the original work showing this in rodents was generated by this department. We learned that this also happens in non-human primates, and you can, these are just serial sections showing you that most of the aberrant sprouting was in the infrapyramidal lobe of the dentate gyrus, and here it is quantified, and you see that following ECT, this IP blade shows significant increases in mossy fiber sprouting compared to anesthesia alone sham. The second measure of neuroplasticity we looked at is cellular proliferation using bromodeoxyuridine incorporation into dividing cells. And you see here the sham animal in the dentate gyrus is not showing much of this, but after ECT, you can count these BRDU stained cells and find quantitative evidence of an increase in these numbers of newly dividing cells induced by ECT, which is these two bars, significantly increased compared to anesthesia alone sham. Now, that's monkeys. What about people? We'd love to have a non-invasive measure of neurogenesis in the brain in humans. A growing body of evidence has been using non-invasive imaging to study morphological changes in the brains of actual patients receiving ECT. Here's an example of one such study that looked at the volume of the hippocampus on the y-axis, showing an increase in that in a dose-dependent fashion. As you increase the number of ECT treatments the patients received, there was an increase in the volume of the hippocampus. Now, we don't know whether that's related to mollusc fiber sprouting or neurogenesis or cellular proliferation of another type, or perhaps other changes that can cause morphological change. Nevertheless, it is compelling and suggests to us we need to understand what's going on here that might be related to mechanism. So I've tried to lay it out in the intro to my talk why we should be interested in ECT. ECT is a moving target. It has evolved over the past decades since its use in psychiatry. I'm going to breeze through about 80 years of progress very briefly to get you up to the current status. The first innovation in ECT was the use of anesthesia, which protects the body from the risk of injury from the motor convulsion. The second innovation was the modification of where the electrodes are placed on the scalp. It used to be everyone got bilateral ECT with two large metal discs placed on each temple. Then we moved to unilateral ECT shown here, and that simple change of moving where the electrodes are placed on the head resulted in dramatic sparing in terms of the adverse cognitive side effects of ECT, in particular amnesia shown here. I'm showing you from one of my studies amnesia for personal events and amnesia for impersonal events. And you can see that both types of memory, the degree of amnesia is substantially reduced when we switch from bilateral to unilateral ECT. Why might that be? I now come to the first aspect of precision, which is spatial precision. Here I'm showing you realistic head modeling of the electric field induced in the brain by bilateral ECT on the right and unilateral ECT on the left. The hot colors show you stronger electric field induced and the cooler colors lower amounts of electric field. And this is the scale here is volts per centimeter. So I'm not showing you, this is not an fMRI or a PET scan. It's actual simulation of the electric field being deposited in the brain. You can see with bilateral, it is truly bilateral, sort of tracks along these white matter tracks here. It's stimulating broad areas of the brain. Unilateral is asymmetrical. It's certainly more on the right hemisphere, but some parts of the left hemisphere are also stimulated. This suggests to us that by controlling where the field goes in the brain, we may be able to reduce the adverse cognitive side effects of ECT. So that's the aspect of spatial dosing. The next innovation in the field of ECT was the aspect of temporal dosing, and that is actually modifying how long each electrical pulse lasts. That's called pulse width. Here you see standard brief pulse ECT, which each electrical pulse, which are square waves, lasts 1 to 2 milliseconds. When you shorten that by a quarter or a third, so you're doing 0.25 or 0.3 milliseconds, again, we dramatically reduce the cognitive side effects. So here you see the brief pulse. This is the same amnesia score. Here I'm showing you for unilateral electroplacement and bilateral electroplacement. When we switch from brief pulse to unilateral, which are the lighter colors here, we again have a substantial decrease in the amount of amnesia. Why might that be the case? Well, neurons respond to specific temporal dynamics. In particular, the chronaxis, which is a measure from the strength duration curve for using electricity to stimulate mammalian neurons, teaches us that ultra-brief pulses are closer to the theoretical optimum for stimulating neurons. So it means once you've stimulated the neuron, you don't want to continue to stimulate it during the time that it's in the refractory period and not able to respond to the stimulation. That excessive stimulation may be contributing to adverse effects. The next innovation in the field of ECT was individualizing dosage. And now we begin to get to the personalized part. We want to have a way of individualizing the doses for patients. And the first approach to doing this was age-based. If you were 70 years old, you would get 70% of the maximal stimulator output. Not very precise because 70-year-olds are quite different. So an innovation was to titrate the seizure threshold for each patient. This was from one of our randomized controlled trials where patients received either a 50% above, 150% above, or 500% above the individually titrated seizure threshold, which means you stimulate with successive trains until the person has a seizure to establish their threshold, and then you give their treatment dose at a certain percentage above that. And you can see in the green bars, which are showing efficacy here, which is response rate, as we increase the dose above the seizure threshold, we're increasing the response rate with unilateral ECT to the point that it actually matches the response rate with bilateral ECT. The red bars are an amnesia scale. Amnesia also increases to some degree as we increase dose, but coupling this individualized seizure threshold titration with the unilateral electrode placement allows us to achieve the same efficacy of ECT, but with cognitive sparing. So I'm building up to, we're doing a lot better with ECT, but we have quite a ways to go. Notice here these red bars, they're not zero. ECT, even unilateral and even ultra-brief pulse, induces amnesia. It's not zero. So in order to move beyond the current best practice, it would be helpful if we had some neurobiological understanding of what was driving the antidepressant effects and the cognitive side effects so that we could design a more targeted, personalized therapy which could improve the dose-response relationship here. And this provides the rationale for the rational design of modern seizure therapies, which would be truly teaching the old dog of ECT a new trick. And let me just explain briefly what I mean by a rational approach to designing device-based therapies. The conventional approach is to take a device, off-the-shelf device, take a set of parameters, run it through a clinical trial, open-label trials, randomized controlled trials, and over a period of, in the case of ECT, decades, we've learned a lot about dose-response relationships that then went back to revising the stimulation protocols to improve outcome. This is a slow and costly process, trial and error process, not really driven by a theory necessarily or mechanistically informed theory. The rational approach is to say, let's insert a few steps here before we get to the randomized controlled trial. Let's understand what the dose delivered to the brain really is by doing in silico experiments and ex vivo models where we can understand where we deposit the electric field. Let's look at in vivo studies in animal models where we could understand how to calibrate this to neuronal thresholds, how to individualize pulse amplitude and optimize the temporal aspects of the train. Let's do in vivo safety studies in animals and in first-in-human trials to understand the tolerability and whether these modifications are improving safety at the expense of anything else that might be related to efficacy, and interdigitate these, revise the protocol before we get to the stage of randomized controlled trials. This was basically the design of our NIMH-funded series of R01s where we took this approach to try to have a rationalized design seizure therapy in a non-human primate model, and I'm going to show you some of the results of that. But first, let's talk about what our targets might be. I'm talking about how to refine and target the thing, but what are we really aiming for? That's where we need the neurobiological evidence. And indeed, it's great to be working in the field today because we do have emerging evidence of potential targets that might be related to therapeutic response. Here is one of them that's looking at the degree of change in depression responding following ECT related to one of these biomarkers, which is change in connectivity of this hippocampal brain area with the rest of the brain. And this change in hippocampal connectivity was related to antidepressant response. So that could be a target. You might try to target your field there or target your seizure spread there. However, with conventional ECT, we have very little control over where we're stimulating. Look again, this is bilateral ECT, this is unilateral ECT next to the target, and you can see we're stimulating much broader brain areas. We needed a more, this is sort of a blunt tool to dissect something that is more anatomically circumscribed. Just to show a little bit more, we've done realistic head modelings of different electrode placements. Here you see bilateral, bifrontal, and right unilateral, the current density in the scalp, and then the stimulation strength relative to the neuronal threshold, which is the amount of electric field needed to stimulate neurons. And this really tells the story here on the coronal sections. Unilateral is not. It really is bilateral stimulation. You can see we've calibrated the scale here so that at threshold stimulation is just this aqua blue color. Everything that's red is more than 3 times above it needs to be to stimulate neurons. To quantify that for you, here I'm showing you on the y-axis the volume of the percent of the entire brain that's being stimulated above the neuronal depolarization threshold. It's almost 100% with bilateral, bifrontal, and unilateral. We've simulated some experimental electrode placements using conventional ECT electrodes. Not much better. We can get down to about 40% with this frontal medial experimental electrode placement, but still it's much higher than it needs to be. Using these realistic head models, we can measure regions of interest and ask how much are we getting in key regions of interest, like the hippocampus, orbital frontal cortex, and so on, in the right and left hemisphere with the different electrode placements, bilateral in the pink and red here, and unilateral in the light green and green. And then we can look at what might be some of our therapeutic targets. For example, the rostral cingulate or subcallosal cingulate here, and I'm showing you for 5 types of ECT. The amount of electric field induced here is all above threshold, so we're hitting that target. But what about some potential non-targets? Now, some might argue that the hippocampus, given its role in memory and plasticity, might be a non-target. I'm in that non-target camp. And here you see that all 5 electrode placements are still stimulating the hippocampus above threshold for neuronal depolarization. It has been theorized that tetanic stimulation of the hippocampus, so remember when we give ECT, it's a repeated train of electrical stimuli. Tetanic stimulation of the hippocampus might induce and saturate long-term potentiation and might be related mechanistically to the adverse cognitive side effects of ECT. If we had a focal tool to induce seizures that could either stimulate hippocampus or not stimulate hippocampus, we could test that hypothesis. Now we have that tool, and as I will show you. This hypothesis has come from, well, first of all, there were classical studies by Stewart and Reed that had posed this as early as 1993. More recent work in animals has verified that ECT does indeed reduce long-term potentiation. Here is the long LTP induced in control rats, and you can see it's suppressed following electroconvulsive shock in rodents. ECT also prolongs the action of long-term depression. Here's control long-term depression that wears off with time. With ECT, it is sustained. So indeed, it does this in animals. Does it do it in patients? Well, and we really need to know that if we want to understand whether it's mechanistically involved in the amnesia-inducing effects of ECT. We don't have a direct measure of that in patients, but back to the volumetric changes, which might be related to changes in plasticity in the hippocampus. This is a small study. It needs to be replicated in more patients, looking at the relationship between hippocampal volume on the x-axis and cognitive function on the y-axis. Most of these lines are going down. The 2 lines that are going up, that's actually the wrong direction. This is the time it takes to do the trail-making task. So all of these measures are showing a significant relationship. More hippocampal volume increase, worse cognitive performance. Today we have physiological measures using transcranial magnetic stimulation to induce evoked responses from the cortex. That's the TMS-EP paradigm. And there has been a study before and after ECT in humans using the TMS-EP showing that a course of ECT increases the amplitude of particular peaks on the evoked potential. This might be a clue to whether ECT is also inducing cortical potentiation in humans, but that needs to be replicated. So conventional ECT is not just stimulating almost the entire brain. It's also inducing very broadly generalized seizures. This is high-density EEG, topographical EEG, before, during, and after an ECT-induced seizure with unilateral ECT. And I'm showing you, this is the 2-channel EEG trace. Those of you who do ECT, this will look familiar to you, the baseline EEG, the high amplitude spiking, the slow wave oscillations, and then the postictal shutdown. The hot colors here on the topo plot show you a very broadly generalized high amplitude seizure. So unilateral ECT is not unilateral, it actually is generating broadly generalized seizures. These broadly generalized seizures have broad effects on glucose metabolism and metabolic activity in the brain. This is a PET study demonstrating that with broad patches of changes in cortical function with bilateral, unilateral, and bifrontal ECT. So our goal for this rational design, trying to head towards personalized therapy, is to achieve the unparalleled efficacy of ECT, but without the amnesia. And if we were to accomplish that, at least in my little part of the field, really would be the holy grail. So we turn first to magnetic stimulation. Transcranial magnetic stimulation, which you all have a great expertise in here, uses electromagnetic induction to pass through the scalp and skull non-invasively and induce small electrical eddy currents in the surface of the brain. It can be more precisely targeted than directly applying electricity to the scalp and skull, because when you apply electricity directly, as with ECT, it is impeded. The scalp and skull are great insulators, impeding the flow of electricity. That doesn't happen with TMS. So TMS is non-invasive. It stimulates superficial areas of the brain. We can get more focal with it than we can with ECT. It avoids the tissue impedance. The idea is to use magnetic stimulation to induce focal seizures, and that's why we coined the phrase magnetic seizure therapy, or MST. The idea is that by enhancing our control over where we're depositing the electric field, this may allow us to gain better control over where the seizure is spreading, and this may allow us to achieve that goal of efficacy without the side effects, enabling focal seizure induction in targeted regions of the brain and trying to avoid those non-targets that might be related to adverse cognitive effects. So it couples the focality and non-invasiveness of TMS with the powerful efficacy of seizures. The underlying hypothesis that motivated the rational development of MST was that by getting more focal electric fields in the brain, we may be able to induce more focal and well-controlled seizures that could spare regions of the brain that are non-targets from the maladaptive plasticity, which might be mechanistically linked to the adverse cognitive side effects, and that this could result in superior cognitive function. This is a four-part hypothesis, and I'm going to give you some data supporting each of these. And this is the spoiler alert. I think we did teach the old dog of ECT some new tricks. So a little bit of a historical side note before I get into this. So when I came to Columbia in 1995, as you heard, for a postdoctoral fellowship, my project was to try to make MST happen. You know, figure out how to use a TMS device, induce seizures with it, see if it can work at first in animal model. And as I was toiling away as a lowly postdoc, I was scooped. It was published in 1996. Someone scooped me, published the first magnetic seizure therapy in a human, no less. And here I was starting with monkeys. And this is an actual quote from that first publication. And it was a pretty accurate report of what I was trying to do. It was like an ECT suite. You use the ECT type anesthesia, methotrexate. You use a bite guard to protect the teeth. Induces a seizure that lasts about 30 seconds. And the advantages over ECT were striking. Yeah, I don't know about this changing clothes part, but anyway, this was published in the, well, it wasn't actually in a medical journal. This is a science fiction novel written by Peter Hernan, and I don't know why there's a bleeding scalpel on this, unless to say this is really this is the bleeding edge of innovation in science. And so when this book came out, I thought, well, I don't know how they got that idea, but it's fun to be trying to take science fiction into science fact. And so here we go. Let's start with the electric field. In order to understand what we're doing with the electric field, it requires that we collaborate with engineers who understand electricity, electrical engineers to be specific. The second part for neurophysiology, working with basic neurophysiologists to understand direct recordings of cortical activity, partnering with neuroanatomists to be able to measure and quantify the neuroplastic effects in the dentate gyrus shown here, and then collaborating with neuropsychologists and experts in cognition in non-human primates and also patients. So this has really been a translational journey and an awesome way to learn how to do science and have a career in science. So let's start with the electric field. The goal here was to design a device that could overcome the anticonvulsant action of anesthesia in order to induce focal seizures in people. And we started with animal models, and it turns out I wanted to work with rats, but tiny coils heat up when you expose them to high voltage electricity, and the smallest brain that we were able to do this in was actually a non-human primate. But it was a real pleasure to work with leaders, pioneers in the field of TMS. This is Tony Barker, Reza Jalanos, and Ian Freeston. Barker, Jalanos, and Freeston were the three authors of the Lancet paper that introduced the first TMS device in the mid-90s. And here I am standing in the middle of them. I wasn't actually on that paper, but it was awesome to be working with them. So in collaboration primarily with Reza Jalanos, we were able to basically soup up a standard TMS device by doubling the number of charging units. And this was our first animal MST device that we developed in 1998, published in 2001. The human device had even more booster modules, kind of a scary looking thing. I kept it behind a curtain. Then we were able to induce a higher dose device that could increase the number, the frequency and train duration that we could deliver. And then this is now the human production model from one of the manufacturers. There are now two manufacturers that make this. So it's really been an evolution in terms of the technology, the type of output it can generate. So I learned from engineering not just their importance in developing these devices, but also their importance in understanding the impact of the device on the brain, thinking of the brain as a system, and being able to develop tools to quantify what we're actually delivering using electric field modeling to visualize the electric field flow in the brain. And I already showed you some of those examples. And it was important to do that if we were able to demonstrate that we were actually sparing the hippocampus. And so here are my collaborators on the engineering part of the project, Angel Pirchev, Wan He Li, and Zi Deng. So these are electric field models that were from Juan's PhD dissertation, which I co-mentored with Angel. So on the top row is ECT, and the bottom row is MST. And here you're seeing the electric field relative to neuronal activation threshold with unilateral ECT. MST, you can see this just very faint sort of ring of tissue just at the surface of the cortex being exposed to stimulation that's just right at the neuronal depolarization threshold. On the coronal section, you can see it a little bit better, very superficial, and really not stimulating the hippocampus, and contrast that with ECT. So we can achieve more focal stimulation with MST. Here we're looking at the MST with the circular coil, which is the blue column here, compared to the 5 forms of ECT, much less in terms of the whole brain. And here we're looking at the hippocampal region of interest, dramatic reduction. So MST we're able to demonstrate stimulates only about 4% of the total brain volume and spares the hippocampus from E-field exposure. So E-field, check. Now let's move on to seizures. He wanted to demonstrate the feasibility of seizure induction with MST in a preclinical model and ultimately in humans, and contrast the quantified expression of the seizures during the seizure and after the seizure between these two interventions. And my neurophysiology collaborators were Charlie Schroeder and Bruce Luber. We first started with the proof of concept inducing the first seizure recorded using scalp EEG in non-human primates. This was published in 2001. Then working with Charlie Schroeder, who surgically implanted intracerebral electrodes in the brains of these monkeys, we were able to record the voltage induced in the brain and the ictal expression, or the intracortical recording of the seizures, with ECT in the red bars compared to MST in the blue bars, showing substantially less E-field in the frontal cortex and no E-field in the temporal lobe with MST compared to ECT. And seizure spread, the seizure did spread the hippocampus in the monkeys, but much less lower amplitude than with ECT. We conducted a randomized controlled trial with MST in the non-human primates using scalp EEG, showing significant differences. This is just a tracing showing that the MST seizures have lower amplitude and less post-ictal suppression. This is using high dose as well. We looked at different aspects of the, in terms of the spectral frequency bands. All of them were significantly less induced with MST than with ECT. We also looked at autonomic response using heart rate. I'll show you that in a little more detail. So we found that the seizure expression with MST was weaker and more focal than with ECT. Here we see ECT on the top and MST on the bottom. The colored balls here, the hot colors show stronger seizure expression throughout the brain, including the hippocampus. With MST, it's really more superficial in the frontal lobe here, with less spread to the hippocampus. Here are humans with the topographical EEG, with unilateral ECT on the top and MST on the bottom. So even in humans using non-invasive recordings, the seizures are weaker and less well generalized. So moving then from seizures to what do these seizures do to neuroplasticity in the hippocampus. Collaborating with neuroanatomist Victoria Rango and human neuropathologist Andrew Dwork, we conducted studies looking at this, first of all, from the safety, because we had not done this in humans yet. So we did do a neuropathological examination of the brains of the monkeys following high doses of MST before we ever did it in a human, and found no evidence of neuropathological damage, not only with MST, but also with high doses of ECT. And I think this paper is important to teach us about the safety of ECT as well. And here we see a stereological cell counting of neurons and glia in different regions of the hippocampal subfields, so CA1 and CA2, 3, with ECT in the red bars, MST in the blue bars, and anesthesia alone in the green. And you can see that there was no reduction in either type of cell in any of these brain areas. So ECT does not cause brain damage, in case you were still wondering about that. Another measure that we looked at was activation of glial cells as measured by GFAP. And ECT does increase the glial activation, which is this blue shading here compared to the sham. MST did not do this. We also counted cells that stained for neuropeptide Y, and ECT actually reduced this, which is in the red bars, in different parts of the dentate gyrus. MST did not reduce it relative to sham. Mossy fiber sprouting. So I already showed you that ECT increases that. MST did not. So here's ECT in two different studies increasing mossy fiber sprouting compared to sham. MST of a low dose did not increase mossy fiber sprouting, but higher dose in the second bar did. So we do see dose-dependent effects on this measure of plasticity. Cellular proliferation using bromideoxyuridine, I already showed you ECT increases that. Both low and high dose MST had absolutely no effect on cellular proliferation. So now we have a seizure-inducing tool that does not give E-field exposure to the hippocampus, does not increase the proliferation of cells in the hippocampus, and at low doses does not increase mossy fiber sprouting. We now have that focal seizure-inducing tool to ask whether these neuroplastic changes are related to cognitive outcome. And we're going to get to that. I'm going to skip over the anesthetic changes so we can get to that. Let's get to cognition. So the goal here was to develop since I was starting in monkeys, a neurocognitive battery for monkeys that would be sensitive to the amnestic effects of ECT, and then to use that battery to contrast ECT versus MST, and then move to the human model where we could use classical neuropsychological tasks. And my wonderful collaborators on the non-human primate studies were Herb Terrace, who's really a leader in the field of primate cognition, and leader now in the field of clinical neuropsychology and effects of ECT neuromodulation, Sean McClintock. So we started monkeys and developed a battery of tasks using a touchscreen monitor where monkeys see different images on the computer screen and have to touch them in a particular order to get a banana pellet reward. And this is just one of the figures of 3 different tasks. ECT, the increasing time means worse performance with ECT in the red bars, significantly worse compared to MST in the blue, which did not differ from anesthesia alone sham in the yellow. We looked at a higher dose treatment, so giving higher doses did not worsen cognitive outcome. You see that ECT shows significant slowing down in performance of this task. MST did not relative to sham. Then looking at humans, this was the first human study where we contrasted in a randomized controlled trial ECT versus MST. I'm just showing you one of the measures, which is speed of reorientation with MST in the blue, much faster than with ECT in the red. Speed of reorientation right after the seizure is correlated with amnesia, and I'm going to show you in the next slide other measures of amnesia as well. Here again is the speed of reorientation, much slower with ECT in the green compared to ECT in the blue compared to MST in the green. And here are some 3 other measures from our large neuropsychological battery showing that ECT impairs verbal memory, phonemic fluency, complex problem solving, whereas MST did not have those effects. We're not the only group to find this. Other groups have replicated this and also found cognitive sparing. Here you see pre-post ECT reductions in a particular neuropsychological task not seen before and after MST. So more focal fields, more focal seizures, less aberrant neuroplasticity in the hippocampus, cognitive sparing, all that's great, but does it work for depression? If it didn't, we wouldn't have achieved much. These are work from Sarah Kaiser and colleagues in Germany, independently showing in a randomized controlled trial that MST had actually no difference in the response rate compared to ECT. In fact, here it looks a little bit better, but that was not significantly different. And here's remission rate, not significantly different. Here are our data in a study of 75 patients with MST in the blue compared to ECT at 6 times above the seizure threshold with unilateral ECT, response rate and remission rate not different. Now there are a number of groups across the world that are doing studies with MST. I'll point out 2 of these groups, which is the CAMH group in Toronto with Jeff Deskalakis and the UT Southwestern group. Here is Mustafa Hussain, but this is actually, the photo was taken in Cardiff. But the UT Southwestern group together are doing a non-inferiority trial contrasting ECT versus MST, which is currently sponsored by the NIMH. That's called the CREST trial, and we anxiously await the results of that. So I'm going to leave MST for a moment, because to do MST, it does require a special device. It requires special electrical requirements, and it would be great if we could just do it with an ECT device. Could we make ECT as focal as MST? Get MST-like results out of a regular ECT device that you already have? Perhaps, but we can't do it with conventional ECT, and let me tell you why and what we're doing about that. So I would say it's time that we re-examine the convention. Why do we do conventional ECT the way we do today? So one of the conventions has to do with our selection of frequency. It turns out that the frequencies used clinically in ECT are inefficient. We know this based on our NIMH-sponsored rational design of seizure therapy study. We iteratively induce seizures with a range of frequencies from low to high, and here I'm showing you the seizure threshold, the number of pulses it took to induce a seizure. You can see it's a U-shaped curve. The red is ECT and the blue is MST, remarkably similar. There's a sweet spot in this curve. It suggests that these lower frequencies, like between 10 and 20 hertz, were more efficient in inducing seizures. So why do we continue to use higher frequencies with conventional ECT? Maybe because we didn't have these data until now. So the theoretical optimum was really around 16 hertz. What we clinically use is much higher. The other convention is the pulse amplitude. Pulse amplitude with all the commercial ECT devices are 0.8 or 0.9 amps. Why? I've shown you that this conventional pulse stimulates broad areas of the brain. Turns out, if you were just to lower the pulse amplitude, this is trivial from an electrical engineering point of view, just put a knob on the device and turn it down. It's like putting the dimmer switch on this plot here. You can see that you get much more sparing of hippocampus if you just dial down the amplitude. Why don't we do that? Convention. Dose titration. I told you that we dose titrate by doing seizure threshold titration. I was taught that as being a very important innovation. Indeed it is, but this is what it looks like. You give a short train, a longer train still, but they're all at this high amplitude that is titanically stimulating the entire brain, and in particular the hippocampus, inducing and saturating LTP, possibly impairing memory. Furthermore, using a fixed high dose for all patients fails to compensate for individual differences in anatomy. We demonstrated that using, again, our head modeling of the electric field induced in the brain. Here I'm systematically varying the volume of the brain from low to high, the scalp conductivity from low to high, and the skull conductivity from low to high. And on the y-axis is showing you what it does to the electric field distribution relative to the threshold for stimulating neurons. And the red lines show you that as you modify these anatomical parameters, we're dramatically shifting the amount of electricity that's getting into the brain. Does this explain inter-individual differences in adverse cognitive side effects of ECT? Well, if it did, we would sure want to gain control of this, and indeed we can, by individually titrating pulse amplitude. For example, if you were to start at low amplitude, moderate amplitude, high amplitude, until you get a seizure, that might be a smarter way of individualizing ECT compared to the conventional approach. It could minimize the volume of brain exposed to excessive field strength and thereby adjust for individual differences in anatomy. And so we have been pursuing a novel approach to doing MST-like work with a regular ECT device, and we've termed this, just to be, lack of a better word, individualized low amplitude seizure therapy, or ILAST. So we're coupling the individual amplitude pulse titration with using lower amplitudes in order to spare hippocampus, And we're using multi-electrode arrays to better shape where the field is going. So instead of using just 2 large electrodes, we can take a page out of the playbook of other fields of neuromodulation, like transcranial direct current stimulation, which is increasingly moving to high definition TDCS, where you can use multi-electrode arrays to better, so this is conventional ECT. Here's a multi-electrode array unilateral ECT, which is much more focal. So now you can see we really can make it more focal. And to bundle that, working with an industry partner, into an adapter that would plug into a conventional ECT device so that we could build it and share it and let other people plug it into the equipment they already have. This, if it worked, could speed dissemination. Okay, so we started with a proof of concept study in animals, and this was part of our NIMH-funded rational design of seizure therapy study, and tried to see if we could induce seizures using this ILAST approach in non-human primates, and indeed we were able to induce a number of low-amplitude seizures. We experimented with a couple of different electrode placements and designs, and we could induce seizures with remarkably low currents, as low as 0.07 amps. We then moved to a human proof of concept study in just 5 patients, an open label trial, using the lowest amplitude that we could give with the current conventional device, which was 0.5 amps. And this just showed that we were able to induce seizures. It doesn't prove anything other than feasibility. You can get seizures with less electricity than typically is used, so that just demonstrated feasibility. We then went back to the animal model, and I'm just showing you our pipeline for doing realistic head modeling in the non-human primates. There's not off-the-shelf packages for this like there are for humans now. So the tissue segmentation, then you model the coil, you fit this together into a finite element mesh or FEM model. We take into account the influence of white matter, the differences in connectivity of white matter. So we did DTIs and incorporate that into the model to generate an electric field distribution. Now we'd like to calibrate that electric field with physiological response in the actual animals that we're using. And so we did do that. In vivo, we measured motor threshold by stimulating with the electrical pulses to determine each monkey's own neuronal depolarization threshold, so that we, and also their seizure threshold, so that we could calibrate our E-field models for neuronal depolarization threshold, not just using a textbook value for that, but the actual animal's threshold. So here's the protocol. We titrated motor threshold with each of our electrode placements. Here I'm just showing you the 2 electrode type, and we also did MST in this study. And so in the field of TMS, motor threshold is well familiar to you. You give single magnetic pulses, measure the thumb twitch, use that to titrate the intensity. The field of ECT really wasn't doing that. The field of ECT was giving trains of stimulation to induce a seizure. So the innovation here is we said, let's take a page out of the playbook of the TMS field. Let's just give single electrical pulses and measure the twitch in the paw, and that's what we did. And we asked whether this corresponded to the electric field induced in that brain area we were stimulating, and I'll show you that correspondence. We also did the conventional seizure threshold titration so that we would know what that would be, but we titrated unconventionally in the amplitude domain. And you can see that this is just showing you a spherical model of that. And so that corresponds to the electric field strength necessary to induce the seizure in that individual. Here I'm showing you the realistic head modeling of our 4 subjects, Chaplin, Marley, Dillon, and Rosenkrantz. And we did 4 different electrode placements on each of them. And here we're just panning through the electric field at their threshold. And you can see there are significant differences across the animals, and in the rows, and also differences across electrode placements in terms of how focal the field could be. I'm going to quantify that for you here. So this is the percent of the entire volume of the brain stimulated with each electrode placement when done in an individually titrated fashion. So individually titrated unilateral was able to induce seizures by exposing as low as 19% of the brain to a suprathreshold field strength. We asked whether the individual pulse amplitude titration might be less variable than seizure threshold titration using the conventional approach, which does not take into account differences in anatomy. On the y-axis here is the coefficient of variation, so it's a measure of how variable the thresholds are with the ILAST in the blue and conventional ECT in the red, and indeed doing this pulse amplitude titration resulted in less inter-individual variability. I was really excited by this finding, and I'm looking forward to seeing if we can replicate this in humans. And that is that the motor threshold measured with single electrical pulses, which is shown in the x-axis, strongly predicted the seizure threshold on the y-axis. The R-squared here is 0.8. If we look at the predictors of seizure threshold we have in clinical ECT, such as age, gender, and electrode placement, these account for less than 40% of individual variation. This is accounting for a much higher percentage of the individual variation. It's really exciting to us because we would like to have a way of predicting seizure threshold without actually inducing a seizure. If motor threshold can do it, that would be exciting. We asked whether the electric field strength in the motor cortex could do it as well, and indeed it did. On the x-axis here is the electric field strength relative to threshold in the motor cortex, and on the y-axis here is the titrated seizure threshold in each subject, and that had an R-squared of 0.99. So we're really excited to see whether just getting an MRI scan at baseline could be enough to predict how much electricity your patient would require. Where we are now is we're doing a first-in-human study in the intramural research program at the NIMH on ILAS to try to translate the findings we had in the non-human primates. We're trying out different electrode placements, each having a more focal or less focal E-field distribution in the brain to learn what is the safety and feasibility of this. We're using physiological and neurocognitive outcome measures and measures of safety as a precursor to then a clinical trial. So I'm moving towards the conclusions here, is that the rational design of precision seizure therapies gives us unique scientific opportunities to probe mechanisms of action, asking whether these differences might underlie the antidepressant and anti-suicide action of one of our most potent and fast-acting treatments we have available today. And if successful, these personalized seizure therapies could offer the promise of a safer, more effective option for our most severely ill psychiatric patients. And indeed, this is an area that I'm committed to as a clinician scientist myself, that the more that we can remove the barriers that prevent patients from receiving the life-saving treatment that they need, the better able we are to serve the patients who are depending on us for their care. These are the members of the non-invasive neuromodulation unit in the intramural research program at the NIMH. And you notice there's a little gap here. And I should mention Valeria, who's a former member of the field and now a member of Yale. But the gap here is we are recruiting. Your picture could be here. John's going to be mad he invited me now. We're recruiting for a medical director for our neuromodulation research program. NIMH as a whole is also recruiting. We're recruiting for what's called health scientist administrators or program officers. Program officers get to do awesome things like give out money for a living and design and think about where we should be investing scientifically. And there are unique windows of time when we're able to recruit, and we're in one right now. So we're recruiting specifically for a new program officer for our geriatrics and aging processes research branch, also for our neuroimaging program. So this is a very important program. Our current director of this has announced her retirement, so we are recruiting an expert in imaging, especially fMRI, PET, MRS, MEG, to join us. And here's how to apply. And also, those of you who are submitting grants in the area of neuromodulation, Dr. David McMullen, I recruited to lead our new program on neuromodulation. He plays an important role not only on the NIMH neuromodulation program, but also is now a team lead in the BRAIN initiative and is a point of contact there. Let me pause here, and I'm happy to take questions. Thank you.